Hello, everyone. We're giving everyone a few seconds to join from the waiting room. Hello, everyone, and thank you for joining. This is Sabrina Paganoni, and as always, we are here for our weekly platform trial webinars. And as we have every week a good team and guest speakers, and I'm here as always with our patient navigation team, Catherine Small and Alison Bulat, as well as our administrative assistant, Jessie Rosenthal. And tonight, we are especially excited to welcome a panel of distinguished guest speakers who will be providing updates on the CNM AU8 clinical development program, and also we focus on expanded access. So with me, I have Dr. Jinsy Andrews from Colombia, who will be the main speaker, and she's here with Dr. Karen Ho from Clene, as well as Dr. Abigail O'Connell from Synapticure. So without further ado, I'll turn it over to you, Jinsy.
Thank you so much. It's a pleasure to join on the webinar to give the update on CNM AU8. I am speaking on behalf of a whole team, actually, since there's multiple parts of the development program. I'm pleased to have Dr. Karen Ho with me from Clene and also Abigail O'Connor from Synapticure, who will also help during the Q&A portion. So if we go to the next slide, I wanted to just kind of touch on a few things. First, I want to just do a quick review of the mechanism of action for CNM AU8 and talk about some of the completed clinical trials and some of the clinical outcome review. Of course, there's been many questions, even in the previous webinars, about the Clene Columbia Synapticure NIH-funded EAP, and I'll try to give an update on that, as well as plans for potentially the global phase 3 clinical trial called Restore ALS. And so it's a lot of territory to cover, but well, of course, you can go to the next slide. We wanted to make sure we provided you with as much information as we can at this time point. Now, this is just a refresher, probably familiar to everybody here, about expanded access, the definition for expanded access, which is a potential pathway for a patient with serious or immediately life-threatening disease or a condition to gain access to an investigational medicinal product for treatment outside of a clinical trial when no comparable or satisfactory alternative therapy options are available. So sometimes in our community in the US and even externally, people will interchangeably use the word compassionate use, but formally the mechanism is referred to in the US as expanded access. If you can go to the next slide. ACT for ALS, you've heard a little bit about it during these webinars, but just as a refresher, ACT for ALS stands for Accelerating Access for Critical Therapies for ALS, and it was signed into law in December of 2021. And it's an important initiative that has several components. There's a granting program for NINDS, and this grant program's intent is really to support scientific and clinical research utilizing the data from intermediate-size expanded access programs with an investigational drug for ALS. And so the goal is really to kind of catalyze and innovate the development of and increase access to potential new therapies in ALS. And I'll just say one additional point here is that while we allow for that access, we're also trying to gain knowledge about ALS, knowledge about biomarkers, knowledge about outcome measures, which can hopefully make us more efficient in formal clinical trials in the future. So we can go to the next slide. And so first, I'll cover a little bit about the mechanism of action for CNM AU8 and also the clinical outcome review. You can move to the next slide. Now, CNM AU8, people affectionately in the clinic, I just had clinic today, refer to as the gold nanoparticles or liquid purple. But it's a mechanism that's really interesting. CNM AU8 is a surface catalytic agent to improve mitochondrial function. And as many of you may already know, mitochondrial function is very critical in ALS. So it's, for those of you that might be familiar with drug development, it's not a small molecule or a monoclonal antibody, but it's a crystalline form of gold that allows NADH to convert to NAD. And it allows for that without the use of any cellular machinery. So what it does is it allows for increased levels of NAD. And when you increase the levels of NAD, you improve mitochondrial function, and this can enhance neuronal function. So it's thought that through this action of increasing NAD, it can improve mitochondrial function and have neuroprotective effects in ALS. So we can go to the next slide. And so Clene has really gone through a number of studies and embarked on a number of late-stage clinical trials and open-label studies, as well as expanded access programs. And they've really tried to evaluate the safety and efficacy of CNM-Au8. Now, the first two studies, Rescue ALS, HEALEY ALS studies, or HEALEY ALS Platform Study, were two randomized, double-blind, placebo-controlled trials evaluating the safety and efficacy signals for CNM-Au8. And there is already an expanded access program that Clene has committed to generate more data from real-world situations. And I think one of the key things here is that Rescue ALS was a 36-week randomized controlled study. HEALEY ALS Platform was a 24-week double-blind randomized controlled study as well. And essentially what we were able to see were preliminary signals in the 30 milligram dose across a broad ALS patient population. If we can go to the next slide, I'll talk a little bit about the clinical outcomes. And so there's been evidence derived from these controlled, placebo-controlled double-blind studies that show that there's a delay in the time to clinical worsening in ALS. And so in the phase 2 study, Rescue ALS, this was the 36-week study, there was a delay to time to ALS clinical worsening. And on this slide, the definition is provided, which is the first occurrence of death, tracheostomy, assisted ventilation, or feeding tube in the ITT or the intent to treat population. Similar findings of delaying significantly the time to clinical worsening was also seen in the HEALEY ALS Platform Study, evaluating the 30 milligram dose of CNM-Au8. Again, delaying the time so the people who were treated had a delayed time to clinical worsening as compared to placebo, which is the dashed line. If you can go to the next slide. This is a slide summarizing some of the findings of the survival data that was evaluated. And so in Rescue ALS, there was up to a 75% decreased risk of death through 168 weeks. So there was the double-blind placebo-controlled portion. And after that placebo-controlled portion, there was an open label extension. So we have observation periods up to 168 weeks. And what we were able to also see was that there was a greater than 90% risk reduction of the death at 24 weeks. And so these signals actually help support further development of Clene in a confirmatory larger clinical trial. So if you go to the next slide, one of the interesting things that we were able to evaluate was neurofilament light chain, which is a key biomarker of neurodegeneration. And so the 30 milligram dose really did show a reduction in plasma neurofilament levels, and it significantly reduced them compared to placebo at 24 weeks. It also demonstrated significantly reduced neurofilament levels at 76 weeks relative to placebo in the long-term evaluation or observation period. And the greatest effect was seen in the 30 milligram dose in delaying the occurrence of clinical worsening events in the highest risk participants based on baseline NFL. So meaning higher neurofilament level is a higher risk population. And so 30 milligrams seemed to have more of an effect in the highest risk participants. If you can go to the next slide. This is just summarizing that we have evaluated CNM-Au8, the 30 milligram dose, in multiple populations, the early to mid stage population in Rescue ALS, the mid to late stage population in the HEALEY ALS Platform Trial. And of course, we're trying to get the observational data from real world experience by providing the expanded access protocols. So again, this reiterates some of the results that I've already talked about in terms of the survival data from those particular studies. So if you can go to the next slide. One of the nice things and special things about CNM-Au8 is its safety and its information that we have of over 500 years of exposure and no real safety signals across populations that include ALS, multiple sclerosis, and Parkinson's disease. So it has clean toxicology findings, it's well tolerated, and we've got pretty good data. Actually, we have very good data on patient exposures across multiple neurodegenerative conditions. If you can go to the next slide. I kind of gave a high level introduction to some of the clinical trials that have completed and some of the safety data that shows really good safety and tolerability. But I did want to talk about the NIH-funded EAP, for which Clene is collaborating with Columbia and also Synapticure on the NIH-funded EAP program. If you want to go to the next slide. There's been a number of questions about the EAP, and I just wanted to take a moment to talk about the NIH-funded program versus the already existing expanded access program that precedes it. The intermediate expanded access program that Clene has been overseeing and was ongoing at prior to the NIH-funded program is really intended for access to the investigational product and really to observe safety and tolerability in a more advanced population. The NIH-funded project is also to allow an increase or in addition to the participants that are already participating in the other expanded access program. So this is a different program with additional slots for participants to gain access to CNM-Au8 in an expanded access pathway. However, it does have research questions built in, and so there are things that we will evaluate in addition to the safety and efficacy in this population. We'll also collect information about biomarkers. We'll get clinical outcomes and try to answer other research questions that are embedded within this NIH-funded mechanism. So just to go over it, the objective is to provide access to the investigational product for approximately 100 participants and that are diagnosed with ALS and not otherwise eligible for clinical trials. And it's intended to evaluate the safety and efficacy of CNM-Au8 in ALS participants in this expanded access program. The treatment period is about 144 weeks of active treatment, so it's a lengthy period of access, and we anticipate that CNM-Au8 will be taken orally or by feeding tube, and the visits will be conducted on a recurring 12-week basis. So we hope one of the unique features about this NIH-funded program is really to strike a balance between conducting visits on site and conducting visits remotely, or maybe even trying to test the feasibility of doing a virtual EAP program as well. So in that vein, Clene is partnering with Synapticure, a specialty telehealth clinical care provider, to help be the virtual site for remote enrollment. And we'll see how this works out. So we can go to the next slide. All right, so again, like I mentioned before, we'll be monitoring safety and efficacy. The safety is being collected just traditionally in terms of collecting the blood work during the visits, and also collecting individual reports of changes in health and adverse events. Efficacy data will not only look at overall function and overall survival, but also breathing assessments, focused questionnaires, blood collection, even biomarker data collection, as well as medical and safety assessments. If you can go to the next slide. I just wanted to give an update on where we are right now. So we are currently trying to operationalize the entire trial process. So Columbia and Clene and Synapticure are working very closely, almost on a daily basis, meeting on a weekly basis, in trying to make sure that we have all of the necessary items to try to get approvals necessary and drug in the right places to try to get that going. We're hoping that we can enable participation from all 50 states, including remote and rural areas, especially by using the virtual telemedicine neurology clinic through Synapticure. And we're hoping that we can get participants enrolled by spring of 2024. And we'll continue to keep everybody updated and communicate proactively as we get closer to enrollment. In addition to this new EAP, Clene will continue to conduct its currently ongoing expanded access program as well. So these programs have enrolled over 250 participants since 2019. So this NIH-funded project is in addition to the existing programs. And if you can go to the next slide, I think this is one of the special things that make this project a little bit unique, is that we really wanted to try and make this as accessible as possible. So we did partner with Synapticure, which operates a 50-state virtual ALS clinic, and they have a multidisciplinary team that actually has a neuromuscular subspecialist and all the necessary subspecialists that can help provide care virtually. And Synapticure will also try to recruit participants who are unable to access EAP through traditional brick and mortar sites. And so if we can go to the next slide, this is just another way of expanding Synapticure's, or explaining Synapticure's role in this program. So essentially they'll be able to support about access for 50% of trial participants. And so the launch date, we'll try to communicate with all participants across our community, and we'll try to establish a framework to support patients with limited access to ALS clinical trial sites. And we're hoping, this is actually one of the aims, one of the other aims in the study is trying to develop this infrastructure for this remote virtual type of enrollment for EAPs. If you can go to the next slide, I'll just go into just quickly the phase 3 program so that we can take some questions here. So if you can go to the next slide, sorry. The phase 3 study is a randomized double-blind placebo-controlled trial in early symptomatic participants on a stable background therapy to reduce mortality and clinical worsening events in ALS. So it's called Restore ALS. I can't take credit for that name. But Clene plans to do a global trial looking or evaluating 30 milligram dose of CNM AU8, which will be randomized 2-to-1 across the globe, involving North America, Europe, and Asia Pacific region, and the Pacific region. If we can go to the next slide. Of course, the plans are still in motion, and the idea is to look at the primary endpoint of survival, which would be the time to death or permanent assisted ventilation. But the global phase 3 intends to also look at delayed time to ALS clinical worsening events, as well as survival and permanent assisted ventilation, feeding tube, and tracheostomy. And so those are more traditional endpoints, but more is coming. The design is still in the planning phase, and it's not yet final. Of course, Clene plans to partner with the community, including clinicians, researchers, and people with lived experiences to kind of guide them through this planning process in the future. And if we can go to the next slide, in 2024, the priorities for CNM AU8 and ALS is really to advance regulatory agency engagement, hopefully to launch the global phase 3 Restore ALS, and to collect data and report on the long-term CNM AU8 experience in ALS. And hopefully the real world experience will help to support all of the other data that will be collected in formal clinical trials, which includes the expanded access programs. And of course, Clene, on behalf of Clene, on behalf of myself and Columbia, and also Synapticure, we like to thank the participants and their families and caregivers in the clinical trials, without whom the studies would not have been possible. Of course, the clinicians and staff who've run the Clene trials, the open label extensions, and especially the preceding expanded access programs. Those are really labors of love and trying to do what's right for people living with ALS. And of course, we want to thank the people living with ALS and their caregivers who've really catapulted ACT for ALS, who've actually allowed for this NIH-funded EAP to get off the ground, and the NIH and NINDS, who's really has been supportive through this process, through the funding process and getting us off the ground, and of course, MGH for this opportunity to share the data. So thank you. And so I think with that, we can take some questions.
Thank you. This was a fantastic presentation. We have, great plans for 2024. So thank you so much for reviewing all of that. And I think really this is, was really a goal of the platform trial, to be able to partner with companies that then can move on to future studies like the phase 3 trial that you are describing. So one question that was asked is, what will be what's the population that will be enrolled in the trial, in the phase 3 trial? I know that it's still in kind of a work in progress. So I believe you probably don't have the final eligibility criteria, but I just wanted to hear your thoughts also in terms of, how that relates to the EAP.
Yeah, so I can defer to Dr. Ho to talk about the maybe any thoughts. I don't know if that's actually finalized, the inclusion exclusion criteria. I don't think it is.
Yes, I'm sorry to say that we don't have that information at the present time.
But I think this would be something that probably in the planning process, having people with lived experiences providing their input and guidance to Clene might be helpful.
That's great. And so there's a question about the endpoints for the phase 3 trial, because you mentioned the primary endpoint of survival. Does it mean that the trial will be longer than other studies like the HEALEY trial, for example, that had a primary endpoint of function adjusted for survival?
I think one of the things that we learned actually from some of the prior clinical trials is that we learned some of the pharmacokinetics of CNM-Au8 and sometimes when we anticipate the effects to occur. And so we've learned that through the open label extensions and the EAPs, but maybe Dr. Ho can explain a little further about maybe the length of duration or maybe considerations for that.
Well, one model that we are considering for the Restore ALS trial is a time to event model rather than a fixed time point in which we would have to take out the trial either quite a long way or enroll many, many hundreds of participants. So a time to event model means that as soon as we arrive at enough of data points to allow us to conclude that there is either futility or efficacy for the drug in terms of survival, then that trial will be stopped and the results will be then read out.
And do you think that ALS functional rating scale or ALS-FRSR will also be one of the endpoints evaluated?
Yes, I believe that is part of the plan.
Okay. Thank you. There's a couple of questions, I'll lump them together, about where the trial will be available. So one question is specifically about New York City. So I'll defer to you, Jinsy, on that. And the other question is also how global will it be? So maybe you can provide some perspectives, both local and global.
In terms of the expanded access program, Columbia will be a site for New York City. We're the lead institution for the NIH-funded expanded access program. The folks that are already participating in the expanded access programs at other sites, it's anticipated that you would continue in those programs. The NIH-funded program is meant for additional slots for people who haven't had an opportunity to participate. As sites become activated, we will be proactive about notifying and probably doing another updated webinar to speak to the sites that are available. And if there is no site that is close by that people can go to enroll in an EAP, then in this particular EAP, then that's the whole point of including Synapticure and allowing remote access for those that may not be in an accessible area. I have to say that the NIH is very interested in making sure that we are inclusive and diverse in the population, and that includes geographic diversity.
And in terms of the phase 3 trial, you mentioned it will be global, and there's a question about the fact that different regions have different standards of care. So do you think that there will be a barrier?
So yeah, go ahead.
We're actively talking to lead clinicians in all of these continents and countries who are specialists in ALS and who are very familiar with running clinical trials, in fact, in running global clinical trials. And so we are actively coordinating the efforts, and hopefully that will help mediate any differences in standards of care that will influence the interpretation of the trial results.
Great. There's a few questions. I'll ask them together in terms of the Regimen C platform trial results testing CNM-Au8. And I know that the publication is in progress, so hopefully that will be available for the entire community to see soon. But for now, can you tell us if there was anything more about the results? You know, were there specific subgroups that responded the best, or what was the main result? Was it on function or survival?
I can defer, to Dr. Ho for the results from the HEALEY platform.
Sure. The very exciting results from the HEALEY platform trial were that on the 30 milligram dose, there was a concordance of actual agreeing data that suggests that we both have survival as well as delayed clinical worsening associated with the 30 milligram dose of CNM-Au8. And the reason why I say concordant is that the HEALEY platform trial was a phase 2 clinical trial that was also conducted. There was another clinical trial, also phase 2, also randomized placebo-controlled, called Rescue ALS, in which we saw very similar results on both survival and delayed clinical worsening. So it's very encouraging to see agreeing results from 2 independently conducted phase 2 clinical trials on CNM-Au8 with the same dose.
Yeah, and that's why also I think it's compelling data to do another trial, like the phase 3 trial that you are planning, so that you can confirm these results, which obviously is critical. So question specifically about the molecule, does it get into the brain? Does it pass the blood-brain barrier?
That's a fantastic question. We have conducted some brain imaging studies called REPAIR. So it was REPAIR in multiple sclerosis individuals as well as REPAIR in Parkinson's disease diagnosed individuals. And what we do is we give them oral doses of CNM-Au8, just the same as we would give oral doses in our Restore ALS or in our phase 2 programs. And what we find is that when you give these doses orally, you can see correlative changes in brain energy metabolites in the expected way that we would expect a favorable change in brain metabolites to occur. So using these special scanning, it's called 31-phosphorus magnetic resonance spectroscopy, we can take a brain scan at the very beginning of the trial before individuals have started taking the drug, and then we can take a brain scan at the end of the trial beyond 12 weeks is basically how we designed the trial. And we were able to show favorable changes in brain metabolites. So that implies that there is brain target engagement that by CNM-Au8, even when it's taken orally. So it gets into the blood, goes down into the stomach, into the bloodstream, and from the bloodstream gets to the brain where it can make these changes, favorable changes in brain energy metabolites.
There's a couple of questions about eligibility for the expanded access. So if somebody is currently in a clinical trial or the open label extension of a clinical trial, whether it's a platform trial or another trial, could they also participate in the expanded access of Clene?
I think I can take this one. The one of the criteria for the so it depends, and I think one of the key inclusion criteria or exclusion criteria is that the EAP, the NIH-funded EAP, is really targeted to individuals that are not currently clinical trial eligible. So if you're in the clinical trial, then you're technically eligible, and you're not eligible to participate in an expanded access program. The other thing I will say, just generally, people get very excited about investigational therapies, and you want to layer them on or off-label prescriptions or maybe over-the-counter supplements. And there are some unknowns, and some people are willing to take the risk. But when you take 2 different investigational therapies, it can make it hard for us to figure out which therapy is helping you or which therapy is not helping you or hurting you even. So typically, clinical trials generally will have an exclusion that you can't take another investigational product concurrently while you're participating in a formal clinical trial. So it's important to talk to your physician or your healthcare provider that's overseeing your care during your participation in a clinical trial to figure that out.
Great, and I'm also happy to say that we will continue to have this team on our webinars. I know Dr. Andrews will be back in March to provide updates. So we're receiving a lot of questions, and I think they're all great. And we definitely want to continue to bring the experts to you so that you can continue to ask these questions live. And I know that many people also watch the recording. So thank you to all of you for following either live or when you have time to watch these webinars. Dr. Andrews and team, do you have any final remarks before we adjourn?
No, but I'm really appreciative of the time to try to give the update as best as we can, you know, at this point in time, but we're working very hard day and night really to try to get everything off the ground. And I'll defer to Dr. Anderson or Dr. Ho, who, if you have any other additional comments.
No, thank you so much.
Yeah, none other that we're very excited to be part of this.
Excellent. And so I know that you have more questions. This team will be back. Catherine, is that March 15th when we are planning this?
14th. So the second
14th.
Thursday in March, we'll have Dr. Andrews back for an EAP discussion.
Fantastic. So please send your questions to Catherine and Allison if you want to send them ahead of time. If not, we'll see you here again in just a few weeks for more discussions about CNM-Au8. Thank you so much.
Thank you.
Thank you.